All right, let's just jump right in. We are facing a massive global health crisis. Our best weapons against bacteria, antibiotics, are starting to fail. But what if the solution isn't a new drug at all? What if it's a virus? Today, we're going to look at how scientists are engineering these incredible viral assassins called phages to hunt down and destroy the world's most dangerous superbugs.
And look, that's not just some scary headline. We are seriously staring down the barrel of a post-antibiotic era, a time where a simple infection, something we don't even think about today, could become deadly again. This challenge, this global problem, is forcing us to get creative and look for some radical new solutions. And it turns out we're finding them in nature's own bacteria killers. I know it sounds totally sci-fi, right? But this strategy has actually been around for over 100 years. The idea is to use bacteriophages, or phages for short, which are these tiny viruses that have evolved over billions of years for one single purpose: to infect and kill very specific bacteria. I mean, they're the most abundant biological things on the entire planet, and now they're getting a serious high-tech upgrade. This slide just breaks it down perfectly. On one hand, you've got the antibiotic approach. It's like a carpet bomb. It wipes out everything. The bad bacteria, sure, but also all the good, beneficial bacteria that we need. And on the other hand, you've got the phage approach. Think of it as a precision strike. It goes in, takes out only the enemy, and leaves our crucial microbiome completely intact. But that amazing specificity, well, that's also its natural weakness.
Okay, but here's the catch. Bacteria aren't just sitting there waiting to be attacked. They've been in this constant evolutionary arms race with phages for, well, eons, and they've developed some really sophisticated defense systems. They've got these molecular scissors that can chop up foreign DNA, and, you've heard of this one, a powerful adaptive immune system we now all know as CRISPR. These defenses can stop a natural phage right in its tracks.
And this is where things get really, really clever. Scientists are taking the bacteria's very own weapon, CRISPR, and turning it right back against them. By arming a phage with a CRISPR-Cas system, they're creating this delivery vehicle that doesn't just infect the target bacteria, it carries a payload designed to absolutely shred the bacteria's DNA from the inside out. So, while the bacteria's own defenses are looking for foreign DNA, the phage's CRISPR system is programmed to target the bacteria's own essential genes. The attack becomes basically unstoppable.
So, once you've armed these phages, this engineering opens up two main ways to attack. One strategy is designed for permanent changes, and the other is for more of a temporary, controlled strike. This slide lays out the two main playbooks brilliantly. Over on the left, we have integrative phages. These guys are like sleeper agents. They slip their genetic code right into the bacteria's chromosome to make a permanent change, like, say, turning off a gene that produces a toxin. Then on the right, you've got non-integrative phages. They act more like a classic virus. Get in, deliver a payload, for a quick, targeted kill, and then they're gone.
Now let's dig into that second group for a second, because these non-replicative phages, they are a real game changer. They're engineered so they literally can't make copies of themselves. Think of them as one-and-done delivery drones. This gives doctors an incredible amount of control. You get a really precise dose, you're not accidentally killing off good bacteria, and you dramatically lower the risk of that engineered phage spreading or swapping genes where it shouldn't. It is all about control and safety.
Okay, this all sounds great in a lab, right? But does it actually work on real people with real infections? Well, a breakthrough clinical trial is giving us a resounding yes, and it's taking this tech from the lab straight into a real-world treatment for some really stubborn urinary tract infections.
So here are the details. The trial is called Eliminate, and it used a therapy called LPBXE01. Now, this isn't just one phage, it's a whole cocktail of phages, all armed with the CRISPR-Cas3 system, which is especially good at just shredding huge chunks of DNA. The target was drug-resistant E. coli, the main cause of UTIs, which have become notoriously hard to treat with regular antibiotics.
And the results? They were fantastic. I mean, patients treated with this CRISPR-enhanced phage cocktail saw a rapid clearance of the E. coli and a complete resolution of their clinical symptoms. This Phase 2 trial gives us some of the strongest evidence we've seen that engineered phages are a totally viable, effective therapy, especially for infections that have just stopped responding to anything else.
But as with any revolutionary technology, we're not at the finish line just yet. There are still some major engineering hurdles, some key bottlenecks that need to be solved.
This is basically the to-do list for the entire field of synthetic biology right now. First, How do you engineer these super-specific phages to hit a wider range of targets without losing that precision? Second, the big one. How do you stay one step ahead of bacteria, which, are absolutely going to evolve resistance to our new phages? Third, we've got to make the whole gene editing process work even more efficiently inside the messy, complex environment of the human body. And finally, we have to figure out how to make sure our own immune systems don't just clear out the phages before they even get a chance to do their job.
And that really brings us to the final critical question here. The science is solid. We have the blueprint to create these viral assassins, these incredibly precise weapons against disease. The ultimate challenge, though, it's a race against time. Can our ingenuity and our engineering actually outpace the relentless, creative force of bacterial evolution? The answer to that question is going to define the next chapter in our fight against superbugs.